So before I go through one of the examples which Elliot Sober mentions in his book on philosophy of biology as a problem associated with the allele frequency based definition of evolution. So he gives 4 problems and I've illustrated just problem number 1 on the board. Before I go through this, I wanted to just point out and consolidate in your minds that the allele frequency based definition is not a rare concept in evolutionary biology, but it is widespread and ubiquitous in the textbooks associated with this science. So I've taken one textbook from my shelf just to point this out, but this is something you'll find in almost all major textbooks on evolutionary biology. So this is Evolutionary Biology by Douglas J. Futuyma, 2nd edition from 1986. And you can see on page 7 of this book, he goes through the conceptions and misconceptions of evolution, and he goes through a lengthy definition of evolution, and towards the end of the definition he mentions the bread and butter of the definition, which is it embraces everything from slight changes in the proportion of different alleles within a population, such as those determining blood types, to the successive alterations that led from the earliest proto-organisms to snails, bees, giraffes and dandelions. Now notice and pay special attention to this line where he says it embraces everything from slight changes in the proportion of different alleles within a population. So what he's saying here is that the lowest level of detecting evolution is slight changes in allele frequencies. So this is right in the actual molecular level where we detect evolution. So when allele frequencies change, then we know by definition that evolution has occurred. So this is the definition you find in all textbooks. So in philosophy of biology, what Elliot Sober has done is he's pointed out that this definition, although is doable, but we have 4 philosophical problems. So actually they're not really philosophical problems, all of them. Some of them are quite mathematical, and you'll see on the illustration I'm going to describe. So there are some problems with this definition of evolution, and one of the problems he points out is the one I'm illustrating on the board. So what happens when an organism, so let's take human being as an organism, selects a mate. So the mate selecting of organism is known as sexual selection, is not random. So a person selects from his own type. So usually you'll find a Pakistani, for example, marry a Pakistani woman, a Bangladeshi will marry a Bangladeshi woman, and so forth. So population genetic models want to be as realistic as possible. So this kind of sexual selection in population genetics or in genetics in general is known as assortative mating. So you have random mating, that a person, an individual organism chooses a random mate, and the likelihood of the female mating with any random male, so any male is equal, so all males have equal opportunity. So that's called random mating. And then you have the assortative mating, where this is where the female will choose an organism, which is a female will choose a mate which is similar in phenotype to herself. So if that's the case, then you will have 3 classes of outcomes. So you have here you have the box represents a genotype, and within the box I've written here genotype. This is zygotes. And then within the genotype you have the allele, so that's an alternative form of that gene on a single loci on a chromosome. So on one locational chromosome you have 2 alternative forms, and one form of the gene, the variant, is known as allele, and then the entire combination is known as the genotype. So here the genotype is homozygous. So homozygotes is when you have 2 alleles that are big A in this case, and so they are matching with each other. So that's called homozygote. So you have homozygote pairs with homozygote, and homozygote pairs with homozygote, and then you've got heterozygotes here pairing with each other. So you have big A and little a pairing with big A, little a. So in assortative mating, you will have a situation where homozygotes pair with homozygotes, so similar phenotype, heterozygotes pair with heterozygotes, similar phenotype. But in a random mating, you have 6 classes of phenotypes. So that's why I specified that it's assortative matings, because there's only 3. And this is the realistic situation in population genetics, because in population genetics you want to model what happens in real life. So Alio Sober brings this example to show that in assortative mating, you have these 3 crosses. If we were to take, for example, 200 pairs, which is consisting of 400 individuals. So this is pair of 50, pair of 100, pair of 50. So in each pair there is, so in pair of 50 there is 100 individuals, in pair of 100 there is 200 individuals, and in pair of 50 there is 100 individuals. So altogether you go 400 individuals. So in 400 individuals which have a genotype associated with them, each one of them, you will have 800 alleles. So each individual has one alternative form of gene known as allele big A in this case, so homozygotes, and you've got allele big A allele and small a allele and so forth. So 800 alleles, 400 individuals, 200 pairs. That's the parental pairs here. So when, let's suppose that these 200 parental pairs, they produce 2 children each. So you have offspring here. So you've got a zygotes here, and you've got heterozygote here, and you've got homozygotes here. So how many do we have of offspring? We've got 100 big A's, homozygotes, plus 50, so that's 150, just the big A's, and then you've got the 100 small A's and another 50 little A's, so that's 150 little A's. So you've got 150 big A's, 150 little A's, plus you've got a heterozygote in the middle, which is 100. So in total you've got, that's going to be 150 times 2, 300 plus 100, that's 400 individuals. And in terms of alleles, these are 800 alleles. So you've got 100 individuals of little a here, to little a homozygote state, with 200 alleles. So each A there has to be counted separately, although the genotypes are 100, the actual alleles are 200. So there are 2 differences. The genotype is the combination of big A, and the actual allele within the genotype is a single A, so you count that separately. So if we count each allele here, so that will be a total of 200 just for this genotype here. So now let's have a look at this example from the allele frequency point of view. So you've got parental pairs that has mated and produced an offspring, 2 offsprings each. So that's quite close to a universal average. People tend to have about 2 to 3 children. So this is a model which is quite realistic. In this situation, you will have 800 allele produced from 800 alleles. But if you take the 800 alleles as parent in the parent pair, the pairs of the parents, you'll find that you've got 100 big A's there, plus another 100 big A's, that's 200. And here you've got 100 A's and big A and 100 little a. So add 100 to 100, that's 200. Add another 100 because there's another big A allele there, that's 300. Add another 100, that's 400. So you've got 400 big A's, and if we take the small A's, you've got 100 little A's here, then another 100 little A's here, and then another 100 little a there and 100 little a. Don't forget this is 100 pair and that's 50 pair. So 1a here is 100 and both combined together here is 100. So this will be 50% of what you will find here. So altogether, if you count all the a's, you will have 400 little a's. So if we do the proportion here, you've got a allele frequency of 50% big a's and 50% small a's in the parental pairs. So it's a 50-50 allele frequency of big A and little a at the parent level. And then when you go down to the offspring, you'll find that the big A's here, you've got 200 frequency plus another 100 here, that's 300. And here you've got 200, but it's only 100 for big A and 100 for small a, the alleles. So the actual genotypes are 100, but 100 is associated with the big A allele, and this 100 is associated with the little a. So you got 200, 100, 100, that's 400 big A frequency, and here you've got 400 small a's. And again, you'll find the frequency is 50% to 50%. So the problem here is that you've gone from the pair of generation 1 to generation 2 without a change in allele frequency. But what has changed? Is there anything that you can see that's changed? There is something that has changed, and that is the genotype. So if we look at the genotypes, so this is AA, big A's, and small a's. What you'll find is here the big A genotype is a quarter, and then the heterozygote state, which is the big A with the small little a, is a half, and then you got the homozygote little a's is quarter. So you got a frequency of genotypes, not allele frequency. Now we're talking about genotype, which is the entire box, is quarter, half, quarter. And if we go into the next generation here, the offspring, the genotype frequency slightly changes. So what you got here is the big A's are 150, so that's 150 big A's, and you got 100 homozygote state, so that's a heterozygote state, and then you got big A's, which are the homozygote big A's, and then you got the little a's, which is another 50 and 100, so that's 150. So if we do the math, you'll find that the little a genotype frequency is 3/8, and then you've got the big A genotype frequency also as 3/8, and then lastly you've got the heterozygote gene frequency as half. So we have a situation where the allele frequency has not changed. It's remained 50-50 and 50-50 in the next generation, but we have a genotype frequency which has changed from being 1/4, half and quarter to 3/8, half and 3/8. So you've got the 3/8, and that's not half actually, that's quarter. So 3/8 and quarter. So 100 would be quarter of the total the total amount of genotypes you have here. So you got 150, then 150 are 300 and 100. So 100 of heterozygote to in a total of 400 genotypes will be 1/4. So the homozygote state has increased from being quarter to 3/8. So 3/8 is larger than quarter, but the heterozygote state has gone from being half to a quarter, so it's gone down. So heterozygote frequency has gone down. So in this situation, from one generation to another generation, we have a genotype frequency. So let's rub this one off. So genotype frequency has gone down for heterozygote, heterozygotes, and it's gone up for homozygotes. So this is the homozygote, the gene frequency has gone up for that, and the heterozygote gene's frequency has gone down. But for the allele frequency, you have no change. Now the problem with this definition of evolution is that you have a situation which is quite a realistic model of population genetics, where the gene frequency allele is also known as gene as well, because it's an alternative on a chromosome, a particular location chromosome. So this gene frequency or allele frequency will be consistent from one generation to another generation, whereas you've got the genotype frequency, which has changed. For heterozygotes it's gone down, for homozygotes it's gone up. So this situation, according to the definition we've just read and all the other definitions, will count as no evolution has occurred. However, we have a genotype frequency which has changed, so it should have been part of the definition. So this is just to identify the problem with the definition based on allele frequencies.